That is really inspirational. So please do that. We look forward to your returning. So we can then just continue with the two presentations that were scheduled before Mr. Petrola's intervention. May therefore request Mr. Javed Iqbal from the DRILS from Hyderabad to make his presentation.
Let me briefly tell you the history of drug discovery for the last 100 years. Having worked in industry myself, when I joined Dr. Reddy's many years ago, I even didn't know that you could design drugs. Well, the drug discovery historically had been based on serendipity, that you make some chemical substance, you try and put it into an animal and see what happens. And then you say, I have discovered a drug. Much later, I think towards the end of last century, with the information technology and all that was happening around it, even the chemists and the biologists thought that they could actually try their hand on designing of drugs. Now, before I really tell you, because it's the most difficult lecture I am giving in my life, I have been a professor in IIT and all that. 8 minutes and you have to tell the audience about what is drug discovery. Thank you, Dr. Desai. Now, just few things that you must keep in mind next 8 minutes that I'm going to talk about. There is protein. I'm sure all of you have heard about protein. Those who have not heard, protein is the main root cause for diseases. And we have thousands and thousands of proteins in our body. Proteins have three-dimensional structures. They are also created by nature, by design. Each protein has its own design and each protein functions in a very different way, depending upon the kind of disease you're talking about. Diseases which are involved in infections, their proteins are very different in design. Diseases that are involved in cardiovascular or let's say lifestyle management diseases, their proteins adopt a very different kind of structure or design. Now what happens really, we drug designers, what we do normally is first try to understand the structure of that protein, which is highly complex, and try and see in a three-dimensional way as to how that protein looks. With the advent of crystallography, information technology, and all that we have seen in the last 30, 40 years, the understanding about the protein structure is very well known now. I mean, I could crystallize a protein and I could say, well, this protein has this kind of three-dimensional structure or design. So what happens really is, as a designer, what I need to do is that I must design a molecule. And when I mean by designing, I first sit on my computer, do some kind of virtual designing or virtual screening as we call it. Then we go to the laboratory and make them actually. Making, of course, is very trivial nowadays because chemistry has become more like a technology. Now, what I'm going to talk about is that first I'll tell you how there is a very long road to new drug. It takes almost 15 years. It sort of costs you, if you're talking about lifestyle management disease, something like $800 million. When we were in Dr. Reddy's, when I was in Dr. Reddy's, we embarked upon a program of diabetes research where we were supposed to bring a drug that we indeed brought to the clinic, but it failed. And I can tell you that most of the drugs that researchers do fail. The one that you see in market actually is the result of a very long drawn battle of almost 15, 20 years with a military-like operation that you see a drug in the market. Obviously all that was happening because people were not using the design principle Serendipity was the name of the game, and therefore you make molecules, you put in animals, and then you say, well, hurrah, this has happened. I got a drug. Now, those days are gone. With the advent of computers and all these technologies that we have, information technology, bioinformatics, and what have you, we now really understand designing a drug in a much better way. Okay, I just told you all this. I don't want to bore you with this because, I just wanted to show you not meant to be read, but I'm just trying to see the complexity that's involved in drug discovery and design. It is actually mind-boggling. It is not something that you can just do at the drop of a hat. It requires tremendous amount of teamwork, lot of patience, lot of failures, learning from the failures, and how do you go about. Each failure teaches you the next step, what you're going to take. Now, what I'm trying to show is that it involves all elements of technology, science, and what have you. There are something like 15 disciplines that are involved in drug discovery and design. We only sort of underplay it because each one of us are so interlinked with the kind of knowledge that we have that no one person is responsible really for inventing a drug. Okay, be that as may, I just wanted to show there's something called cell. I'm sure most of you know. This cell has a membrane that has to be permeated by a drug molecule that we make, and it should get into this. But there are a lot of barriers to this drug molecule entering into the cell because cell is comprising of some very complex structures. That is also by nature. Nature has made by design, and therefore it would not allow everything that you make to get into the cell. And cell is the heart of where every action is taking place. Okay, so one has to understand all these before one really sits down on a computer, look into the complex structure of drug, and try and see if you can design a drug. Okay. Now, there is something called virtual screening. I've already mentioned to you about it. This is nothing but this basically requires, structure-based. I'll tell you a little bit about these things. Let me put first all this. See, if you look into this, there are certain different ways in which you can look into it, where this is a protein structure, which is typically shown here. We take a molecule that we virtually design and put into this and see where this molecule or drug rather sits in the cavity of this protein. Then we have other thing called ligand-based quantitative structure, because if I change the structure of a drug, how it changes the property of that drug towards a particular biological response. Then what we know is that we try and see, then we construct a certain philosophy hypothesis by making a triangle like this. Say, if a molecule fits into this triangle, and if that drug is fulfilling this requirement, then I might be able to get a drug that would actually hit that particular protein target that I want to actually stop from functioning. Then there is something called knowledge-based, which is a chemical biology network, which requires tremendous amount of bioinformatics knowledge that has been unearthed by the protein structure that has been now unraveled over the last 20 odd years, especially after the human genome sequence was unearthed. Okay, I will not bore you with this. Please remind me just 2 minutes before by this thing. See, what a chemist sees. I, as a chemist, trained as a chemist, I would like to see my drug aspirin, which you all know, which is like this. Sildenafil, which is actually a very important drug, and actually it looks like this. Okay, this has become very important nowadays. So, as a chemist, I see aspirin like this, where I have a bond, I have a CH3, I have an oxygen, I have a benzene ring. Similarly, in sildenafil, I see like this. However, protein does not look like this. Proteins, okay, sorry, here, what you do, you convert this into a one-dimensional structure into a line notation, which could be like this. I can simplify this structure by design, and I can put these things like this. There's a model called SMILES. But a protein does not see these like, protein sees my aspirin and sildenafil or Viagra like this. It is a sort of cloud that actually surrounds this, and this is how protein looks at it. Now, what are these red and blue things? You know, every molecule has an electronic structure, a cloud of electron around it. We chemists make it very simple by drawing lines and calling CON. In reality, actually, if you look into a molecule would actually look like a cloud of this kind, where some place it will be negatively charged, some place it will be positively charged. And this is what a protein confronts when it comes across an aspirin molecule or a Viagra molecule. Now, let's not get into this. This could be understood by simply electrostatic fields. If you happen to know a little bit about it, you would actually appreciate what I am talking about. Okay, so what really here is, when a molecule is like this, and when a protein sees this, you can further simplify this by design. Now, what you do is, aspirin now has been sort of compared, it has been segmented into a green and a red sort of area, where green is hydrophilic, water loving, and red is hydrophobic, water hating. Protein, if you look into, protein contains enormous amount of water molecule inside. Protein structure, three-dimensional structure, is actually because of water. And okay, so what I'm trying to say is that water hating and water loving for a drug becomes an extremely important phenomena, and this is completely characterized by designing the molecule in such a way that certain part of the molecule or drug has a property of water loving, certain part of the molecule has a property of water hating, and this is how it would go and sit in the cavity of a protein and block the function of that particular protein. So this is how the protein sees a particular drug like aspirin or Viagra. Now we can then do a lot of other things like QSAR based things and all that stuff, and then quickly I'll show you how we put this into this is a protein, and this molecule in this big one is a drug molecule, and we put into a grid. Try and look into the interaction this has with the protein, and when I talk about interactions, it is electrostatic interactions. Molecule has a certain charge, protein has a certain charge. How these charges are going to complement each other in terms of accommodating that particular drug molecule into the cavity of the protein. So this is how actually you go, and then you can further simplify by design by looking into red area and green area, or yellow and green, which is nothing but these are all electronegative and electropositive areas. Now what we do, last 1 minute I'll tell you how we do designing. This is where designing comes. The background is all protein, you can see. This is my drug. Now I want to design this drug in the environment of this protein, which I can see on my computer. And then what I do what is known as scaffold hopping. I remove the middle part because I think that middle part was not fitting beautifully into the environment of the protein. I do a bit of calculations, bit of things, and I then look into this and see what are the distances between these, what are the donor acceptors and charge and all that. These are things that are part of the design that we do, and finally we bring a totally new part in this, which now is called scaffold hopping, and this basically becomes a new drug. So we started from there, and I'll tell you, in HIV, this was a drug that actually everyone has been talking about. This is the molecule. Now those of you can sort of have a little understanding of this sort of structure, you will realize that this structure is very different from this structure. If you just sort of concentrate on that. And this has been done nothing but by designing the experiment and looking into in such a way that you can completely transform this original structure into this, and therefore these do have similar biological properties but different structures. This has come about by design, and I can show you in the environment of the protein, one molecule of HIV protease is like this, the other one you can see is different, and both work or function the same way but have a slight biological response. Now I would stop here because it was very difficult for me. I thought 8 minutes would be too long, but I think it's all over now, and thank you very much.